Hello my dear students, this is Dr. 𝗵𝗵𝗴𝗿𝗺, your biochemistry faculty. Well, in this video we are going to discuss about a very important and an interesting process, translation. Now, first of all, what is this process, where it occurs, what is the significance of this, everything we are going to discuss now. So, translation is simply, as the word suggests, it is translating the information which we get from DNA in the form of genetic information. Okay. So, actually as per central dogma of information, what happens is that DNA forms RNA, right, by a process known as transcription, and RNA translates the information which is hidden in the form of genetic code by a process known as translation. But after the discovery of the retroviruses, RNA can also be converted into DNA, which is opposite of transcription, so we call it as reverse transcription. Right, and DNA is forming daughter DNA by replication. So, this is how the flow of information takes place. Now, the question is that where this process occurs. So, whenever the information is being translated, whenever the concept of protein comes, one thing we have to keep it in mind, that it is occurring on the ribosomes. Very important. So, if we talk about translation, the site is the ribosomes. Now the question is that the process is occurring on the ribosomes. What is the transporter which is carrying the information? From where the information is being read? So everything is a systematic and a organized way. So there are three types of RNAs which are involved in this process of translation, which are ribosomal RNA, transfer RNA, and messenger RNA or mRNA. Okay, so first of all, if we talk about messenger RNA, messenger means it is taking the messages, that means something is present on it. So it is bearing the codons. tRNA, it is bearing the anticodons, which is carrying a specific amino acid which is coding for that particular codon. And ribosomal RNA is the basically machinery where the entire process is taking place, right. So, messenger RNA bear codons and it is linear in structure. Transfer RNA bear anticodons and it has a clover leaf-like structure, anticodon, clover leaf-like structure. And ribosomal RNA is having subunits. We know that in prokaryotes and eukaryotes, the ribosomal RNA or ribosomes are different. So, in prokaryotes, it is 70S, in eukaryotes, it is 80S. 70S, which is further off two subunits, 30S and 50S, and 80S is 40S and 60S. So, this is the entire infrastructure required for translation. Now, coming over to this, what are the different steps involved in this? And obviously, prokaryotes may it will be different, eukaryotes may it will be different. Because if we talk about the prokaryotes have ribosomes arranged on that mRNA chain, so they are known as the polysomes. And eukaryotes have a special structure. Yeah, you can say ribosomes are different, transfer RNA is different, messenger RNA is different, their synthesis is also different. And in prokaryotes, synthesis is again different by RNA polymerase, which has 5 subunits, right. So, when we talk about translation, first of all, we have to actually arrange the machinery. So, if we talk about rRNA, it has 2 subunits. a smaller subunit and a larger subunit. And this subunit is bearing two sites. A site is the acceptor site, means it is accepting the amino acids, and P site is the peptidyl transferase site. Right. Then if we talk about transfer RNA or tRNA, it has a clover leaf-like structure. Now clover leaf-like structure means this is 5' end, right? This is 3' end. And this 3' end is bearing the activated amino acid. This 3' end which has CCA, it carries the activated amino acid. Activated amino acid. And here this is anticodon. Now this anticodon has to search for a specific codon which is going to express for a particular amino acid. So that is present in messenger RNA or mRNA. So in mRNA, which I told you, it has a linear structure like this. Obviously in the direction 5' to 3' and which is bearing these codons like this. And the most important property which we have to remember is what is the nature of this codon first of all. So this is nothing but this is a genetic code. The information is hidden in the form of genetic code. So what comes under this genetic code? Or what are the important properties of this? If we talk about the definition, we can simply say, as it is clear from the word itself, code is hidden. Why? Because it is dealing with the heredity, because genetic is there. So that code is decoded with the help of DNA, RNA in the form of proteins. So what are the properties of this genetic code? The most important one is it is triplet. Because if it is double, it cannot code for 20 amino acids. Okay, but if it is triplet, it can code for more than 20 amino acids, and that is what exactly happens. So it is triplet as it has to code for 20 standard amino acids. This is important. So it has to be triplet. Then, so first is it is triplet, second is it is universal. Universal means it is applicable everywhere, because again if we talk about the genetic information or genes, it is present everywhere. Now the third important point is it is comma-less. Right, and it is without punctuation. This is also very, very important. It is without punctuation. Right, then fifth point to remember about this is the most important one. It is degenerate. Degenerate. So degenerate means, degeneracy means, जैसे suppose if one say AUG is there, suppose if I say it is coding for say A amino acid. If this AUG में the last base pair is replaced by some other AUC, it might code for the same amino acid. Or even if it is coding for some other, it is not going to make any change. Means degeneracy, a change in the third base pair. This is very, very important. Degeneracy means that there is a change in the third base pair. Right. And since it was discovered by a scientist named 𝗵𝗶𝗵𝗺, it is known as 𝗵𝗵𝗵𝗺 hypothesis. So, change in the third base pair is known as 𝗵𝗵𝗵𝗺 hypothesis. And it will always lead to silent mutation, means it is not going to make any significant change. Right. So, degeneracy of the genetic code is very, very important. 𝗵𝗵𝗵𝗺 hypothesis, or it might lead to silent mutation. Right. So, this is one very important property. The genetic code is triplet. It is universal, it is commaless, it is without punctuation, and it is degenerate. Or it is non-ambiguous also, whatever we can say. And this is the most important point which we have to remember. You can see that degeneracy in the third base pair like this. Now coming over to this, जैसे I told you कि codons are there which are going to be recognized by this specific anticodon, which will be bearing a specific amino acid on the 3' end. And this is how an entire polypeptide chain will be formed, or the protein will be read. Because if we talk about the proteins, so many proteins, we talk about the peptide hormones, we talk about and everything is collagen, they are synthesized in the body. And how they are synthesized? They are synthesized by this process. Okay. So, now coming over to this, what are the steps of translation? First of all, steps of translation. Now, the first step is, if we talk about the 3 steps, I can say there are 3 steps which are present. First step is initiation. Second is elongation, and third is termination. Right? So, initiation is the first step, then elongation takes place, and then finally in the end the chain terminates. Now, first of all we will take initiation. Now, initiation is a most elaborative process, very elaborated or very long process. Right. Now, what happens in initiation? The activation of amino acid is there. Any amino acid which has to undergo the process, it has to be activated. So, activation of amino acid. And obviously, if any amino acid is activated, it requires ATP. So, what will happen is this amino acid will be activated in the presence of enzyme amino acyl tRNA synthetase. So, the enzyme is amino acyl tRNA synthetase, and it is going to form AMP complex, or we can say ATP will be used like this, right. Basically, one ATP which is giving rise to AMP and PPI, so 2 ATP are used. Then second is 2 ATP, or I would just write like this, ATP which is forming AMP. So, it is equivalent to 2 ATP. Now once the amino acid is activated, second is charging of tRNA takes place. Now what is this charging of tRNA? Obviously this amino acid AMP complex will give that activated amino acid to the transfer RNA. So charging of transfer RNA means that this is how the amino acid is transported like this, and rest it is like this, 5 prime 3 prime, and this is the anticodon. Okay. Up now in this entire process we can say that this becomes the rate limiting enzyme, or this is the fidelity enzyme of translation. So I'll just write it here that amino acyl tRNA synthetase is the fidelity enzyme of translation, because it is regulating the entire process. Very important. Now if we talk about in eukaryotes, what happens is, because this process will always require certain initiation factors. So if we talk about the eukaryotes, in eukaryotes what happens is, we know that they have ATS, which means 40S and 60S. Okay. So what happens here is, the first step is simple, you can say that methionine, which is the first amino acid which is red, will combine with tRNA, right, and it will react with this 40S, and in the presence of initiation factors, initiation factor 1, 2 and 3, it is going to form a pre-initiation complex. And this pre-initiation complex, with the help of other initiation factors which are known as FIGA, right, they are going to form the initiation complex. Right, in some books it is written as 43S also. So 43S, when it combines with methionine and transfer RNA plus amino acid is there, it is going to form pre-initiation complex, which will combine with then 60S to form a initiation complex, right. Now once this initiation complex is produced, what will happen is, we can say that the first amino acid which is read in translation, this is also very, very important, the first amino acid which is read in translation is methionine in eukaryotes and formyl methionine in prokaryotes. So, or it is the initiation codon, whatever we can say. So, initiation codon in prokaryotes is formyl methionine, right, and initiation codon in eukaryotes is simple methionine, which is coded by AUG, right. So, we can say that initiation codon is this. So, how the assembly is maintained? So, if I have to write initiation step, this is the smaller subunit, right, this is the larger subunit, this is P, this is A. A will accept the amino acid and RNA, messenger RNA will be like this, right, 3 prime, and first codon is AUG, like this. Now this AUG will recognize only that transfer RNA which is bearing methionine. I mean, in eukaryotes it will be methionine, or in prokaryotes it will be formyl methionine. Later on this formyl group is removed by formylase enzyme, but initially for recognition, for initiation of the process, this formyl methionine is required, right. So if we talk about transfer RNA, this transfer RNA will be like this, and here it will be bearing methionine, and here it will be UAC, I mean the corresponding anticodon. So always remember, AUG is going to code for methionine, A is going to accept. So this is how the initiation starts. Up after this, if I talk about AUG, then maybe AAA is there. So AAA is coding for phenylalanine. So another amino acid will come, or this another transfer RNA will come like this, right. It will be bearing phenylalanine like this. So initiation is just ki A is accepting, or here initiation step requires 2 ATP. So 2 ATP are required for initiation step for recognizing only one amino acid at a time, recognizing one amino acid at a time, right, which is very important. So 2 ATP, because energetics of every process is important. Now second step is, this is what? This is initiation. So initiation mein we can simply say that the amino acid is being activated, or we are generating a complex. Clear? And here I have already shown the two sides. So A is accepting the amino acid. Now the role of P site will come. So second step becomes the elongation step. Now what happens in this elongation step? Elongation step cannot occur alone. So it will always require factors. So elongation requires factors. And if we talk about the factors here, it has, because again prokaryotes may it will be different, eukaryotes may it will be different. So if we talk about prokaryotes and eukaryotes, in prokaryotes these elongation factors are TU and TS and G. In eukaryotes they are simply elongation factors 1 alpha, 1 beta, 1 gamma, like this, right. So we can say that with the help of these elongation factors, whether it is taking place in prokaryotes, whether it is taking place in eukaryotes, it has to start. Now elongation requires two steps which are very, very important. The two steps are, the first step is obviously first step is, you can say, transfer, and second is translocation, right. So always remember, transfer mane, ab jaise suppose if this is the case, so this is the smaller subunit, this is the larger subunit, and we can say that here AUG is there and AAA is there. AUG, AAA. Now, this transfer RNA has methionine, which is the first amino acid to be recognized. On the other tRNA, it has what? Along with methionine, it has phenylalanine. UUU. Okay. Now what will happen is, methionine is here, phenylalanine is here. So methionine has to form a peptide bond with phenylalanine because we require a polypeptide. So this is what is elongation. Very important. So we can say transfer or elongation में there will be formation of a peptide bond. And once it is forming a peptide bond, then it will be translocated. Why? Because now the chain has to move further. Why chain has to move further? Because it has to read another amino acid. So if I say this is the case, after this say amino acid is AUU for example, and this AUU is coding for say lysine for example, right. So what is happening here is कि first of all methionine is read, then phenylalanine is read, and the third amino acid is already ready to be read. So first transfer RNA has to now leave the vicinity of this smaller subunit. Why? Because it has to bring in more and more of amino acids. So that is what is exactly happening, कि there will be a translocation, right. So the enzyme here will be peptidyl transferase. Peptidyl transferase, which is again a ribozyme obviously, it will help in the formation of peptide bond and the translocation. And the translocation will be such that the first red amino acid will always be at the bottom. Again, this is very important. So I can say that peptidyl transferase having a ribozyme activity, okay. So it will help in the formation of a peptide bond, obviously, bond between two amino acids, and then translocation is there. And what is the purpose behind translocation? The first amino acid will always stay at the bottom. Right. So we can say elongation will be there like this. So in all process, one GTP will be required. Clear? One GTP here and one in the first step. So overall I can say that there will be involvement of 4 ATP in the you can say in the acceptance of the entire process. Clear? So now what happens is that in this translocation we can simply say that 2 GTP are required. Right, amino acid is activated for the first time, so no other ATP is required for the activation. A new amino acid, if it is added, then only it will be required. This is only for one amino acid at the one time. Getting my point? Matlab you have to understand this. Whenever we are accepting one amino acid at one time, this is how the process is going on. Later stages mein, this is how the energetics will multiply. If another amino acid will come, again 2 ATP will be required for the activation, 2 GTP will be required for the translocation and for the formation of peptide bond. So the process will keep on going. So I can say that 4 ATP in total, because GTP and ATP are energy equivalence mein same, in total are required for accepting one amino acid, accepting one amino acid at a time. right? So this is how it goes on. Now thing is that elongation will keep on going unless and until there are 3 non-sense codons which appear on the mRNA chain. Elongation will keep on going and it will be like a red like that, or you can say that the third step will be termination. Now termination is what? Like, I'll just write it and then I will explain that the information will be read continuously throughout. Okay? Information in the form of amino acids, will be read continuously right, but when non-sense codons appear on the way, non-sense codons appear, the chain terminates, because these non-sense codons do not have a corresponding anticodon on transfer RNA. So obviously they are not going to code for any amino acid. So information in the form of amino acids will be read continuously, but when non-sense codons appear, chain terminates. And these non-sense codons are UAG, UGA and UAA. Okay, so these are the 3 non-sense codons which when appear on the messenger RNA, they will never be accepted as such because they do not have a corresponding anticodon. Very important. And the chain will be terminated and it will be released. Now here what we have to do is that release factors are required just to release the chain. But peptidyl transferase is the one which is the main enzyme which is going to take out that peptide bond, because what happens is with the help of release factors. So if we talk about the release factors, that release factor 1, 2 and 3, they will be there in prokaryotes, and simply you can say release factor will be there in eukaryotes. So these 2 factors which will help in releasing the polypeptide chain. Releasing the polypeptide chain. And this is how, say I told you methionine will always stay at the bottom, and this is how different amino acids which are red, they will be arranged like this. So this becomes a polypeptide which is released from the vicinity. Okay. So let's quickly revise. Translation is we are translating the information which is hidden in the form of genetic code by forming a protein or a polypeptide. Initiation process is there. Initiation process is the one which is initiating the process. Initiation is the most elaborative process because we are going to make a complex which is pre-initiation complex or the initiation complex, where the subunits are arranged. mRNA is just swirling around the smaller subunit and transfer RNA is being activated by carrying a specific amino acid. So always remember, initiation factor plays a very important role in initiating, because amino acids have to be activated first of all. So amino acids are activated with the help of enzyme amino-acyl tRNA synthetase. Once amino acids are activated, then they are transferred on transfer RNA. So that is why the second step there is charging of tRNA, because T transfer, it is the one which is transferring the information in the form of that. Then comes elongation. Now elongation mein P and A sites are there. A site is which is continuously accepting. P is which is going to form a peptide bond. So it has two activities. First of all, it is going to form a peptide bond, and second is it is going to translocate. Because ultimately we cannot have so many transfer RNAs in the entire vicinity. We need to have only one transfer RNA which will bear the polypeptide and the other transfer RNA which is carrying a specific amino acid. Okay. So peptide bond is produced, then it is translocated. So again for that purpose energy is required. That is why I have written 2 GTP. Then what happens is the chain keeps on going. When we get a nonsense codon on mRNA chain, the chain has to terminate. And the reason behind that is that this nonsense codons do not have a corresponding anticodon. That is why they cannot code for any specific amino acid. The chain will be finished and the polypeptide will be released. Very important, right? Now, this is translation. Now, another important feature about this translation is that if you remember in amino acids we had discussed about 2 very special proteins, selenocysteine and pyrrolysine. There I told you, you have to just remember selenocysteine and pyrrolysine. It is the 21st amino acid and this is the 22nd amino acid. And if you remember, I told you one very important thing, that both are coded by nonsense codons. Just say this is coded by UGA, this is coded by UAG. But still they code for specific, polypeptides, or selenocysteine is considered to be a very, very important enzyme also. It's a coenzyme also. So these two are synthesized by a process known as co-translation. Very important. So we can say that selenocysteine, which is the 21st amino acid, is synthesized by a process of co-translation. This is one very important point to be remembered in translation. Then another important point about this is that we had discussed about 2 apoproteins in lipid metabolism. Okay, वैसे तो lipoproteins की apoproteins, there is a separate classification for that, but we mentioned about ApoB48 and ApoB100. Right, and I told you a very important point about this is that ApoB100 and ApoB48 are synthesized in the body by again RNA editing. So that's also a part of translation. Okay, so this is also a part of translation, or you can say RNA editing or processing is done. Right. So how this process is done, because here I am writing 48 and here I am writing 100. So that means the structure is well elucidated. So here what happens is that the process takes place in the liver and the intestines. So what is happening here is, we can simply say that this is the polypeptide, say this is the liver and this is the intestine. Say DNA, say we can say RNA is being produced like this, and this RNA is having this sequence. Now what happens is, it also undergoes editing, So in the intestines, when it undergoes editing, this C is replaced by U. So what happens is, it is going to form 5' to 3'. Instead of C, U comes. Whereas in the liver, it is like this only. So what happens in the intestine, since C is replaced by U, and we all know that U is what? UAA is a stop codon. So immediately the translation is stopped. So you can say that is why intestines mein ApoB48 is produced, and in the liver ApoB100 is produced. This is the most important point to remember here. So in the liver this is the process, and in the intestines this is going to take place. Okay. So ApoB100 is specific for VLDL and LDL, whereas ApoB48 is specific only for chylomicrons. Right. So this is what is translation. Is it clear? So this is all about the process of translation, where simply we are translating the information hidden in the form of genetic code. In the next video, we are going to discuss about the inhibitors of translation, and we are going to discuss the post-translational modification, because once the polypeptide is produced, it has to modify itself, because transcription also, whenever transcription is over, it has to be modified. Same in translation, once it is translated, it again has to be modified. So that's all in this. Thank you so much.